Let's continue here with more physical factors that are part of your innate immune functions. Ciliary escalator is a good one. That would be the ciliated respiratory epithelium, for example, that traps microbes that you have inhaled and then also trap in the mucus, and then you're coughing it out eventually. So that's you're bringing the microbes basically back up. So the ciliary escalator traps the microbes that you've inhaled, and then you're coughing them back out eventually. Earwax prevents some microbes from entering the ear. Urine cleans the urethra via flow, so the flow is outside of your body and it just kind of washes any bacteria that are trying to migrate up, it washes them out. Also vaginal secretions, they move microorganisms out of the vaginal tract. And then we have peristalsis, defecation, vomiting, diarrhea. So all of these are mechanisms that attempt to get rid of any kind of microorganisms or dangerous substances, toxins, for example. Here is an example for the ciliary escalator. You can see here this little cilia, these little hair-like extensions sitting on top of this epithelium. And you're going to have some mucus, and there will be some trapped particles, and eventually you're going to cough everything out, so move it back up. Now moving on to chemical factors, they would be including the sebum. These are secretions on your skin. They have a lower pH than what most bacteria would like, so pH 3 to 5 of the skin. Lysozyme is in perspiration and tears, saliva, especially in saliva in your mouth, also in urine, and destroys bacterial cell walls, so limits bacterial growth. And then the low pH in your stomach, so the gastric juices in your stomach, very low pH, right around pH 2, that would destroy most bacteria and toxins. And then the low pH of vaginal secretions, so that we have a pH somewhere around 4 that also inhibit microbial growth. Now, a really great defense is actually your normal microbiota. So you are inviting bacteria that are actually friendly to you. They don't mean any harm. In fact, a lot of them, they are doing good for you. You're inviting them in and offering them a place to stay in exchange for them basically outcompeting any pathogens that might want to settle there too and live there. So you're inviting the normal microbiota, microorganisms, and offer them a place to be. You offer them also some nutrition, and in exchange they occupy growth niches that otherwise could be occupied by more dangerous things. And also a lot of times they're producing vitamins for you or doing other healthy things for you. And they also basically your normal microbiota and your immune system, they have a very good relationship, and it helps your immune system to really know what's dangerous and what's not dangerous. So your normal microbiota, very important. It competes with pathogens, stuff that would be dangerous for you via microbial antagonism. Sometimes they can produce substances that are harmful to pathogens, and they alter conditions that affect pathogen survival. So they're really occupying these growth niches that otherwise could potentially be occupied by something dangerous, a pathogen. We have the term commensalism that we had before, that is part of this symbiotic relationship. I really like to think of the normal microbiota much more than commensalism. It's really that we both benefit, and the bacteria benefit, and the bacteria benefit, and we benefit as well. So, but here commensalism means that one organism benefits while the host is unharmed. But really, these bacteria are actually doing a lot more than what you think for you. Probiotics, those are live microbial cultures that you can take in pill form, or even in your yogurt if you have live cultures in there, that are administered or taken as a pill to exert a beneficial benefit. And in recent years, the normal microbiota has really taken front and center stage in terms of benefits for you. So it's a very important part of our defense system, and it does much more than people had originally thought, and we'll talk some more about that. And now let's move on to formed elements in blood. And here we have the cells of the immune system. Of course, the red blood cells, that's the vast majority of your cells that are in the blood. The leukocytes are the ones that are participating in your immune function. So the white blood cells are the ones that are that is your immune system. Then we also have platelets, and they participate with your clotting responses. New blood cells in general, they are created in the bone marrow from a stem cell, the mother of all your blood cells. That's the hematopoietic stem cell, and the process is called hematopoiesis. So here would be the hemocytoblast, that is a pluripotent stem cell, and it can create all of the different white and red blood cells that we have. So here you see the lymphoid stem cell, and it can lead to these T cells, B cells, natural killer cells, and plasma cells and whatnot. So you can see that right here. And here would be your myeloid stem cells that lead to erythrocytes, platelets from megakaryocytes. And then we have mast cells, eosinophils, basophils, neutrophils. So there's the whole variety right there. Okay, so among the formed elements in blood, we have granulocytes, and that would be neutrophils, basophils, and eosinophils. They're called granulocytes because they have cytoplasmic granules, and in order to visualize them, you have to stain them. Neutrophils, they like a neutral dye, and we usually don't offer them one because neutrophils are easy to recognize. They're very numerous, and they have a segmented nucleus, and so you can easily spot them. Basophils and eosinophils, they have both granules, and the basophils, they release histamine from these granules, and then the eosinophils, they have hydrolytic enzymes in there, and they would be going more on the attack against parasites such as pinworms, for example. Basophils, they work in allergic reactions. They really, because of the histamine, and they induce inflammatory responses. Both of them are granulocytes, and if you wanted to stain them, you would have to stain the granules in order to distinguish them. Eosinophils, they got their name from the eosin dye that they like very much, and it stains their granules orange. Basophils, they got their name because they like a basic dye, and it stains them with the crystal violet, they're kind of deep purple. So if you are staining these granulocytes, the neutrophils, they don't pick up a dye, but you can see how segmented that nucleus is. See how it's sort of shattered in pieces. That's very typical of a neutrophil. It's also referred to as a polymorphonuclear neutrophil, PMNs. And it's about 2/3 of your circulating white blood cells are those neutrophils. Basophils, they stain very dark with a basic dye with the crystal violet, and you can see lots of granules inside of them. And then the eosinophils, they're much lighter. They have these orange granules right here. And otherwise, the size is the same between basophils and eosinophils, and they're both of them are pretty rare. And so in order to distinguish these two cells, you would have to stain the granules. And again, the basophils, they like a basic dye, and they stain dark, the granules, and the eosinophils, they like the eosin dye. Okay, moving on to agranulocytes. There we have monocytes. Once they migrate into tissues, they are called macrophages. We also have dendritic cells. They're found in skin, mucous membranes, thymus. They are phagocytic. And then we have our lymphocytes. And lymphocytes can differentiate into T cells and B cells. and also into natural killer cells, NK cells. Now, T cells engage in cell-mediated immune functions. B cells, these guys here, they form antibodies, and natural killer cells, they kind of like the cytotoxic T cells, or the T cells in general, that are engaging in more cell-mediated functions, and they all play a role in adaptive immune functions. So here is a table that summarizes the white blood cells, the types and how often they occur, what they look like, how they differentiate. For example, the monocyte into a macrophage is kind of a good thing to know. And so this is for your review and as you study this. Now let's move on to the lymphatic system. The lymphatic system, of course, has this name association with lymphocytes. So the lymphatic system is composed of the lymph fluid itself, lymphatic vessels, lymphoid tissues, and also red bone marrow. It contains lymphocytes and phagocytic cells, and the lymph fluid carries microbes to lymph nodes where filtering takes place, and but also the lymphocytes and macrophages that are resident populations there, they destroy the pathogens. So we have these immune functions here, and here would be a review of the lymphatic system. Different parts of the lymphatic system are listed here. So the thymus, if you still have one, if you are young enough, we lose it after the teenage years. You're losing your thymus. Then lymph nodes are all over the body. Then the thoracic duct is a very large lymphatic vessel that returns fluids from that were picked up by lymph vessels. Then the spleen, of course, is part of the lymphatic system, and lymphatic vessels, okay, all over the place. So here is also an interesting image that shows you how lymphatic vessels, they start blind-ended in tissues. It's not a circulation, they start blind-ended. So we have a lymphatic capillary that starts sort of randomly in these tissues and picks up fluids that were left over from the cardiovascular system, and then they will basically collect these fluids and drain them into larger and larger lymphatic vessels until you're draining everything back into the cardiovascular system. Meanwhile, you're going through lymphatic tissues, lymph nodes, and filtering everything, making sure that there are no pathogens in there. Okay, so now let's take a look at phagocytes. Phago means to eat from the Greek, and cyte, of course, is cell. So phagocyte is a cell that eats, an eating cell. We have a variety of phagocytes. We have fixed macrophages that are resident in tissues and organs, and then we have free or wandering macrophages that roam tissues and kind of take a look, sort of like a police patrol that patrol a certain area. So here you have a macrophage engulfing rod-shaped bacteria. So that's their job. They're engulfing bacteria and internalizing them and destroying them. Here you have the steps of phagocytosis. So first you need to attract a phagocytic cell to the site where a pathogenic microorganism is. So that's called chemotaxis. Usually there's some chemical signals that will attract the phagocytic cell to the site where it's needed. Then we need to make sure that the phagocytic cell will adhere to a surface, and that it will is able to attach. The phagocyte needs to be able to attach to the surface of the microorganism. And then we have ingestion or opsonization, where opsonization basically means that the pathogen that needs to be ingested has been flagged, and so therefore now a lot of times it's antibodies that are coating a microorganism, and then your phagocytic cell has an easier time, but it's not even necessary. So phagocytic cells, they can figure it out. They will engulf the microorganism and then will ingest it, so engulf it, internalize it, and then digest it by fusing it with a lysosome, making a phagolysosome, and that has hydrolytic enzymes in it, and then we're digesting that microorganism. You have the steps of phagocytosis one more time in a diagram. It's a little bit easier to follow. So first, you need to have chemotaxis and adherence, and that will be the first step. And then sort of this bacterium here was scouted out and flagged maybe, and now this phagocytic cell is engulfing it and packaging it up into a vesicle, and so that will be ingestion. Now we have this microorganism in the vesicle. Now we need to fuse a lysosome. So now we're fusing a lysosome. We have a phagolysosome, and the hydrolytic enzymes, they will digest the bacterium. You can see it's completely picked apart. And then a lot of times the phagocytic cell, like a macrophage, they will spit out the trash, basically. Some pathogens, they have mechanisms by which they can evade the phagocytosis. And so here are some strategies. Some bacteria, they inhibit adherence of a macrophage, for example. They have M proteins or capsules, and this is employed by Streptococcus pyogenes or Streptococcus pneumoniae. Some bacteria, they can kill phagocytes, so they have leukocidins. For example, certain strains of Staphylococcus aureus can do this. Then some bacteria